Okay, so today what we're going to be going through is the practice SAC for the enzymes. I do recommend, if you are not even doing the topic test, that you do make sure that you're doing the SACs, the practice ones that I do put up, or the activities. In the case of the quartz practice SACs, they are extremely similar to the ones that we'll actually be doing. Not so that you can rote learn questions, but so that you understand how they're asking you questions, the style of questions, and then you can have a look as well as the responses they're expecting, so the keywords, the detail that they're expecting, and the comparisons. All right. So it's really important that as a minimum you are making sure that you're going through these SACs and that you're taking the time to do that for yourself. All right. The reason why I've included in this front page, so this is similar to what you'll see when we do our real SACs. So your name obviously needs to go here. It tells you about the outcome, the tasks, which can also be seen on the pages that I put up on Edmodo, the cover pages. But what I really need you to pay attention to here is that you normally have two sessions. So the first session is your practical, and it normally goes for 90 minutes. Now I can hear you saying to me that class only goes for 80. So most of the time, you're right, we're going to have to do some of it out of class time. So it might even have to be before school, might have to be after lunch, before lunch, after school. Okay, so it's not a heap of time after school or whatever, but you need to make sure you are given that extra 10 or 20 minutes so that it's fair, because the rest of the state will begin that same time as well if they're using the same SAC. Okay, so please make sure that you're having a look at those cover pages and make sure you're highlighting those in your planner so that you are there on time. Okay, the second session that you do get is where you get to do your discussion questions. Normally it's about 50 or 60 minutes. There is one I think that you get 40 minutes, but again that's on the cover page. So you should know that before you come in, and I put the time up on the board anyway. So in the first session, the only things that you are allowed to complete is the experiment itself. Okay, and I'll go through how I would expect you to do the experiment in a sec. I'm starting with preparation and then follow through with it. And the other thing that you're allowed to do is your results. So if there's any tables or graphs, you can do that in that first lesson. Okay, what I do expect that you do with your results to make your life a lot easier when it comes to doing the discussion questions in the second half is that you do a summary of your results. So you know exactly what those results mean. Okay, you not have to flick back and forth and waste that time. You understand thoroughly what those results mean to you. Okay, you should know also what you expect to see, and then compare that to your results to see if you had exactly the same thing. All right, beautiful. So this is the stem of the question. Now most of the time what they'll do is they'll give you something that you haven't heard of before, and you might think that it's content that we've missed out on, and you might start to freak out, but no, it's not the case. What it is they'll ask you to apply your understanding. So in this case they're going to ask you to apply your understanding of enzymes to the scenario here that they're given to you of the phosphatase. Okay, so don't be alarmed by the information, just get a highlighter and start to unpack it and summarize. So I'll go through and I'll show you what I mean by that. Okay, so introduction, phosphatase. Before I even go past that word, and I know it's an enzyme SAC, so you do expect to see it, but I want you to start really paying attention to the words that you're reading. So phosphatase ends in A's, so it must be an enzyme. Okay, can I just point out also that not all enzymes, majority of enzymes do, but not all enzymes. So something like pepsin, which is a well-known one and was discovered, it was one of the first ones to be discovered, doesn't follow that rule of ASE. Okay, so majority of enzymes, any ones that you generally haven't heard of, will end in ASE. Okay, so a group of enzymes that are able to cleave to a phosphate group. All right, so I want you to start thinking about this sort of thing. So phosphate, you would see phosphate in ATP, you would see it in a nucleotide, and you'd also see it in a phospholipid. So this sort of basic understanding here comes from chapter 1. So I think it's really important you need to apply it to all of the topics that we cover throughout the year. So if you need to go back over that because you didn't get that cue, make sure you do, please. Okay? So phosphatase, a group of enzymes that are able to cleave a phosphate group. So this, obviously, here is their substrate. They need to bind to that in order to cleave it off. Okay? And they play an important role in cellular metabolism. Now, metabolism is just the total amount of reactions that are occurring within your body and how fast they're occurring. I think it's great. It's a word that we use every day. We say, oh, I've got a fast metabolism, or no, my metabolism's slowing down as I'm getting older. And all that means is that your body is, the rate in which reactions occur in your body is decreasing. That's really interesting. Okay, the free phosphates have a variety of roles in cells, including the formation, here we go, of DNA and RNA, which is your nucleotides, and also found in cell membranes, which is your phospholipid. Okay. Now, the optimal range of the phosphate has been used in this experiment is a pH of 5. All right. Now, whenever you come across optimal range, you probably will be asked to define that, whether it's a topic test, SAC, or an exam. And what you need to be able to say is that that's when the reaction is able to work at its most effective, at its best, so it's most effective. Okay, so that's because it has a decreased amount of activation energy, and also the particles are moving at such a rate that it's allowing for the reaction to happen at its fastest. Okay, so that's important that you know that. Let's change your colour so you can see where I'm going. An extract derived from bean sprouts, yum yum, is used as the enzyme solution. So that's where we're getting our phosphatase from. An artificial substance, phenylalanine phosphate PPP, which we can see down here in the equation, is created by combining phenylalanine, a pH indicator for basic solutions, and I'm just going to change that to purple. It's important. I think you may have come across it sometime during junior science, but it's important that you know that. So in a basic solution, it's generally, and we keep reading, I'm spoiling the punchlines here. So it's created by combining phenylalanine, a pH indicator of basic solutions, together with a phosphate group. The phenylalanine indicator is without colour, so it's clear. All right, so just put that in black. So that's clear, but turns pink in alkaline solution. Okay, so alkaline just means basic. All right. It's a base, in other words, okay, which we got up here. Phenylalanine remains colourless while a phosphate group is attached. So I feel like there was a lot of colours in there. There was a lot of, we're talking about pH, we're talking about phosphates, we're talking about colours. So let's try and break that down a bit. So I'm just going to move that over to the side. All right. So we can see in the equation here we've got phenylalanine phosphate, so PPP, okay, and then we've got the enzyme, so our phosphatase, and then that, remember, that binds to a phosphate group, which a phosphate is here. Okay, once it binds to it, then it's going to break it apart, so then we'll have our phenylalanine and our phosphate. Okay, so let's read through that again. So it says, an artificial substance of phenylalanine phosphate, so we've got that here, is created by combining phenylalanine together with the phosphate. Great. So that's this here again. Phenylalanine indicator is without colour in an acidic solution, but turns a pink colour in an alkaline solution. Okay, so it needs to be in a basic solution, otherwise it's not going to have any colour. So I'm just going to write that up here. Basic solution, we'll get a colour. And what colour do we think we'll get? It's like a pinky purple colour. Okay. Okay. However, the phenylalanine remains colourless while a phosphate group is attached. Okay. So if we get to this side of the equation, because the phosphatase has worked and it's been able to bind to the phosphate group, then we're going to see a colour change. But remember, it does need to be in a basic solution. Okay? If, and I'll change this to a completely different colour, if we do not see, if the enzyme is subjected to a pH or a temperature that is not its optimal temperature and it's not working efficiently, or it's so slow that we can't see it working, this PPP will not turn into phenolphthalein and phosphate. So therefore, we will see no colour change. All right. Or if the reaction is occurring, but it's happening at a lot slower rate, you might see a reduced colour change. Okay. So, see what I mean by the stem of the question is extremely important. Like, you could go and do this experiment, and you might not even know what the results are meaning. And then when you come to do your discussion questions, not only are you having to think about what to respond to, but then you're trying to figure out also about what your results mean as well. Okay? All right, now I kind of scribbled over the bottom bit, but sodium carbonate, an alkali solution, which is basic, okay, is used to end the reaction. It does two things. It stops the enzyme from reacting any further, and it also causes the free phenolphthalein to turn pink. All right, so we are going to, what color can I do? Green. Stop reaction by adding our basic solution. And then if there has been any reaction, okay, we'll be able to measure it by the colour change. Okay, and if we see a colour change, okay, we know that the enzyme has been working, it's been able to cleave to the phosphate groups and remove them. If we see no colour change, okay, then it's obvious the enzyme hasn't worked and it's still back in this state here with a PPP. Okay, if we see a reduced colour change, we know that the enzyme's not working as efficiently because it still does have some of those phosphates forming that PPP molecule. Okay, I hope that does help before we go into the discussion questions. If it doesn't, please make sure you pause it and summarise this yourself. Okay, so you always get given pieces of paper to summarise sorts of things. So you are more than welcome to use a whole book if you like, but it just when we do finish the class, I need to collect it, so you can't take it with you. But it's very important that you do understand what's going on. Okay, awesome. So this is the method, and I've told you time and time and time again, what I would normally do is I would take a I would create a flow chart. Now, I'll see if I can take a photo while I'm recording this. Even though you aren't doing the prac itself, I would still suggest that you do, in fact, hang on, can I take a photo? Okay, so even though you are not doing the experiment, I would still get used to doing a flow chart. Okay, so you can see that I've set it out there with different sections, different highlighted sections. I think I've even put things in there about the results and what it means. So get used to doing that. All right, I suggest you do it. These are results that I did post on Edmodo, and you can see that this is the color chart definitely will help you. So up here, so this is the reaction. It's still ranking pretty much of how strong the color change is. Okay, so remember, you are only going to see color change at the end. End of experiment. When we add a sodium bicarbonate, I'm just going to abbreviate sodium carbonate for them. Okay, because that's the alkaline solution. So don't expect to see a color change until that's added in. Not that you're doing the experiment anyway, but just so you know, okay? If we see it up here, then we would say that the enzyme is working quite efficiently, okay? So it's able to bind to the phosphate group and remove it. And then because it's been removed, remember that the phenolphthalein in that alkaline solution turns into a pinky purple color, okay? If we see something down here, okay, so from about 2 onwards, we're still seeing a color change. However, the color change is nowhere near as great. So you would say that the enzyme is still working, however, it's not at its optimal temperature, okay, and it's happening a lot slower. Whereas at 0, we're not seeing any reaction, okay. So the enzyme might still be working, but we might not be seeing it in the time frame that we're doing it as well. So that's also a really important point to keep, okay. Beautiful. Now, I'm not going to do the graph for you. I think that's a really important skill. I'm going to tell you a few things that you need to include. So one thing that you may or may not have done is line of best fit. And I'm just going to put some random dots on here. Okay, pretty much what a line of best fit is, that half of the dots will be on top of the line and half of the dots will be on the bottom of the line. Okay, so it doesn't need to be extremely accurate. I would use a ruler, but I don't have one in this case. So I'm just going to roughly draw a line through. Okay, so you can see that roughly two dots are on top, 2 are below, and 1 sort of on the line there, okay? And that's your line of best fit. In a case where, in an upcoming SAC, you might need to read off this, okay? So using a ruler is really important. Again, with that sort of thing, don't worry about not doing it like absolutely accurately, because it's going to be from your results, okay? So whenever they ask you to respond based on your results, you need to put a quantitative data, whatever your results are, put it in, okay? But it's always really important that you know about what you should have seen as well. So make sure after you do the experiment, you go home and you do some research on what normally would occur, okay? Because errors and mistakes happen all the time, especially when you're under pressure in the lab, all right? The other thing that you need to make sure that you are including on your graph is, and this is a little acronym that I've started using as a tick list, okay? So, T for title. Please make sure it's a descriptive title. Don't write graph. Nothing annoys me more, okay? I'm going to jump all over the place with it. S is scale, okay? So you need to make sure that you have a scale to tell you on the bottom and the top and the units, all right? So you might have temperature on the bottom, not on this one, obviously. You're gonna have reaction rate on one side. You'd have 0 to 10, like we saw in the results here. So you would use this ranking as one of your axes, and then you'll have the pH buffer on the other one, okay? But make sure that you have units. It's really important. So if you don't have units, I'm not going to give them to you. All right. Labels, even intervals. So make sure if you've got 0 here and you've got 5 here, so you've got 1, 2, 3, 4 spaces, that you're not doing 6 here. Okay. They need to be evenly spaced out. Okay. And you need to have your axes labeled. Okay. With your labels, please make sure you have a key if you have more than one line on here. Don't do bar graphs. Unless it says to you to do a bar graph, I expect that you're doing a line graph. Okay? Line graphs are the way to go. Smiley face. Okay? So please make sure that you're, there's an activity I think I've posted on to Edmodo actually that goes over line of best fit. Please make sure you're doing that, or come to see me and we can have a practice at it. And use tails when you're doing your graphs. Okay, so please make sure you have even intervals. Bring a ruler in. I'm going to write that here because you need a ruler. Alright, you need to have a descriptive heading, you need to have labels, you need to have units, you need to have a key. Alright, awesome.